Well, thank you, everyone, and welcome to our first seminar on National Myeloma Month, a very busy month for Myeloma Australia and our myeloma community. I'm Mark Henderson. I'm the CEO of Myeloma Australia. I'd like to firstly acknowledge the traditional custodians of the country that we all join on right throughout Australia, and pass my recognition on to the custodians of the land, their connections to land, sea and community. I want to pay respect to elders past, present, and extend that respect to all Aboriginal and Torres Strait Islander people today on our call. Thank you for being here with us. It's such a great honour to be part of this organisation. Generally, it's a great honour to lead Myeloma Australia, and it's such an important. The community is so important to the work we do. We've just recently come out of a major event. For those of you who couldn't make that event, we're getting the video out shortly so you could see what went on. But we had an amazing lunch that probably many people watching this would have been at the lunch, and we really putting the voice of myeloma patients and the community and carers out very loudly. I can tell you that people are starting to listen to the messages they're hearing about the importance of understanding myeloma, be aware of myeloma, and to be aware of the opportunity we've got in front of us to bring a cure to bear, which may be a period of time, 10 years or so to make that happen. But we want to fight for the myeloma community, and we see that very much as our role. So I want to thank you for being part of our series, our education series. As you know, that if you ever need any of us and our fantastic nursing team, you reach out and we're keen to support you. So I want to thank you for being part of today. And I'm going to hand over to one of our specialist myeloma nurses, Kath Bowley, who's part of our 18-strong nursing team based in New South Wales. Thanks for being with us, and over to you, Kath.
Good morning, everyone. Thank you so much, Mark, and welcome, everybody. I'm so glad to be here with you all. My name is Kath Bowley, and I'm very lucky to be one of the specialist myeloma nurses with Myeloma Australia. I'm based in New South Wales. I'm joined today by my co-host and fellow nurse from Victoria, Danielle Hovey. Our theme today is Living Longer with Myeloma, with a focus on advances in treatment and access to those treatments in Australia. We do have some wonderful presentations coming up for you. Today's seminar runs for about 2.5 hours, and we will have a short 5-minute break scheduled between the first and second sessions around 1:30 p.m. Sydney time. Working behind the scenes today is our colleague Tash from Queensland. She's available on the nurse support line if you're experiencing any technical difficulties, and that number is 1800 693 566. Tash will also be monitoring your questions, which you can submit today via the Q&A box if you scroll down the page, or via the live chat box, which is available by tapping the 3 dots in the top right corner of your screen. We will do our best to respond to questions today, but please include your email so we can respond in that way if required. You could also email any unanswered questions directly to our nurses inbox, which is nurses@myeloma.org.au. A reminder that today's session is going to be recorded and available for playback on our website, along with the second seminar coming up later this month. We'd love to know what you think about today's session, and your feedback is appreciated so that we can tailor future events to meet your needs. You'll find an evaluation form at the bottom of this webpage. We'd love for you to do that after the seminar. With the housekeeping out of the way, it is now my absolute pleasure to welcome our first presenter, Dr. Peter Millay. Associate Professor Peter Millay practices as a consultant haematologist in clinical and laboratory haematology at the Princess Alexandra Hospital and is Associate Professor with the University of Queensland Medical School. He holds appointments with the Medical Scientific Advisory Group of Myeloma Australia, the Australian Amyloidosis Network, and the National Blood Cancer Task Force. He is a past chairman of ALLG and current chair of the Australian Amyloidosis Network. He heads the myeloma service as well as the Queensland Amyloidosis Centre. We are so grateful he's with us today to present on advances in myeloma treatment in the current Australian climate. Thank you, Peter.
Thanks very much, Kath. So It's now my pleasure to be able to talk a little bit about the role of treatments, particularly new treatments, CAR-T and bispecifics in the current Australian climate. And I'd like particularly just thank one of my colleagues down in Adelaide, Dr. Cindy Lee, who's lent me several of her slides. Particularly, you'll notice the ones that are prettier. So this is what I'm going to talk about. Just have a little overview of where we are at the moment in terms of current therapies, where we've come from. Then talk a little bit about the role of the immune system in cancer, because understanding of this has led to an explosion of a new therapeutic approach. And that is the use of what's called CAR-T cells and bispecific antibodies, as well as antibody drug conjugates in the management of multiple myeloma. And I'll briefly mention some of the other new therapies that are around the corner as well. So what's our goal in treating myeloma? What are we trying to achieve? It's a highly treatable type of cancer. It probably remains incurable. And a few years ago, this slide said it remains incurable, but I'm not so sure anymore. I think there are a proportion of patients that are experiencing incredibly long remissions in excess of 2 decades. And with the introduction of some of these therapies that interact with the immune system, I think the therapeutic outcomes with myeloma may indeed be changing. And so I'm not so sure anymore that it's an incurable cancer. But in terms of practically, what are we trying to do when a patient presents? We're trying to achieve remission, that is to kill as much of the myeloma as possible. We need to manage complications along the way, and that includes complications of the myeloma itself, as well as complications of the treatment. And also important and not to be forgotten is to control the bone disease. And I'll just mention one slide on this because it's a very important part of treatment of myeloma. That is management of bone disease, which still the majority of patients with myeloma have to some degree. In fact, pain remains a very common presenting symptom of myeloma because of the destruction that happens with the myeloma cancer in bones. So getting pain relief is very important, particularly early on, but equally important is over time to wean down that pain relief. Radiotherapy remains a very important treatment modality, particularly if you've got a myeloma bone lesion pressing on something important like the spinal cord. It's very important for some patients who present with their myeloma only in one spot and nowhere else. That's an uncommon situation called a plasmacytoma, or it can be very useful for persistent bony lesions. It's also important for all myeloma patients to be treated with some type of drug to reduce future fractures, reduce the likelihood for need for an orthopedic surgeon to have to come and strengthen a bone. These are drugs called bisphosphonates, of which the 2 most common in use are permethrinate and zoledronate. And not only do they help control myeloma disease, but in fact, patients live longer if they're on a bisphosphonate. So it's an important part of treatment. A more recent treatment available is a different class of agents, but doing a similar job, of which there is one called denosumab. It's approved in bone disease for other cancers, but not yet reimbursed in Australia. So what have we got to treat myeloma? Here's the list of not every myeloma therapy, but the ones we've got available. And if you can see, I've got a little mark next to some of them as to whether they're available on the PBS, that is, they're available to all Australians at no cost, or whether they're only available at the moment through a medicines access program, that is, pharma sometimes give away the drugs either for free or at a price, but they're available, or if they're only really available at the moment on trial. And so we've got these different classes of agents. We've got the immunomodulatory drugs, which are all derived originally from thalidomide, and there's lenalidomide and pomalidomide on the PBS, and there's a couple of others that are probably not too far away, iberdomide, and the drug with a lot of numbers has got a name now, mezigdomide. These are both very potent drugs of this family, and they've got the advantage of being oral therapies, offering convenience. We've got the proteasome inhibitors, of which the first one was bortezomib. There's a version which is a little like oral bortezomib called ixazomib, and then the second generation proteasome inhibitor, carfilzomib, all very active drugs in myeloma. We've got what we can call traditional chemotherapy agents, which are drugs like cyclophosphamide and melphalan. We use melphalan in particular in high doses with autologous stem cell transplantation. Then there's the monoclonal antibodies, of which we have ones directed against CD38, a molecule that's on the surface of all myeloma cells. And those ones are daratumumab, which is available on the PBS, and isatuximab, which is only available in certain trials. Elotuzumab targets something different on the surface of myeloma cells and is available on the PBS, whereas belantamab, which has a drug toxin attached to an antibody, is only available on trial or on compassionate program. Then we've got some other drugs. Selinexor is the only one of these available on the PBS, both in combination with Velcade and by itself. We have T-cell engagers, of which there is one, teclistamab, that's available in a Medicines Access program, but it is expensive. Otherwise, there are numerous trials in Australia of various T-cell engagers. We have CAR T-cells available only on trial at present, and a new class of drugs, BCL2 inhibitors. This is an oral therapy, highly effective in myeloma, but yet to be worked out how to safely deliver the drug. So they're the therapeutic choices that we've got at the moment. And here's the way in which they're used. Up the top in blue, I've got now. This is kind of how we use them. And in the below in blue, the future, how they might be used. So at the moment, there's initial treatment to control the myeloma. And this is, for most patients in Australia, a combination of lenalidomide and dexamethasone, or those two drugs with Velcade as well, the so-called VRD regimen. And for patients who are young, and young in myeloma terms is less than 75, so maybe not what the population thinks is young, but if you're young enough and no significant health problems, a transplant, an autologous stem cell transplant to consolidate that initial response. And then whether you have a transplant or not, some form of maintenance, which is typically lenalidomide at lower dose and continuing until there's problems with side effects or until the disease progresses. And then if the myeloma comes back, we've got a variety of agents as listed on the previous slide, but there are various regimens based on daratumumab, carfilzomib, pomalidomide, selinexor, elotuzumab, in a variety of different combinations. And at this point, treatment becomes much more individualized, depending on things like prior responses, side effects from prior therapy, ability to travel to a center to receive an injection as opposed to an oral therapy. Treatments are very much different between different patients in that setting. In the future, though, what would we hope to get available in the shorter rather than longer term? Well, certainly the ability to get daratumumab in the initial treatment of myeloma in combination with Revlimid and dexamethasone. This is a highly active therapy. It's had a proven role for patients who are not transplant candidates. And it's again before our funders to see if it might become available. In other patients, the ability to have carfilzomib up front may also be beneficial. It may be in future we won't be using lenalidomide as maintenance, but maybe one of the newer agents in that family, like iberdomide or mezigdomide. And in relapse, at the moment, we're hoping to get CAR T cells approved at later lines of therapy to give us another very powerful option, which we'll hear about later on in the seminar. We hope to get some of these newer bispecific or trispecific T-cell engagers that I'll explain a little bit more about. Maybe venetoclax will find its place, but it's very active in particular types of myeloma. So that's kind of where the field's heading as an overview. So just to talk a little bit about one of those therapies, which has been around for a long time now, since the mid-90s, and that's autologous stem cell transplantation. Autologous means your own stem cells are being used as opposed to someone else's, which is called allogeneic. The stem cells are in fact special cells in the bone marrow which give rise to all of the cells in the blood. So your stem cells give rise to all your white cells, all your red cells, all your platelets. And transplantation refers simply to putting back those cells into the body so that they can grow up again. And you can see in the diagram there that the process involves moving those stem cells out of the body, out of the bone marrow where they normally live, to circulate in the blood where they can be collected from patients. They're stored down, they're measured to make sure they are going to work well. And then after some initial high-dose chemotherapy, which is what is the active agent in treating the myeloma, in killing the myeloma cells, the stem cells are given back, because otherwise after that dose of chemotherapy, the blood cells would not recover on their own. So the stem cell transplant is a way of supporting the ability to deliver a very high dose of chemotherapy. And then after a couple of weeks, those stem cells grow back up and one recovers from the transplant. It is, however, an arduous and difficult therapy, but it works, which is why we still utilize it. Just to give you some of the idea of how things have come, in the left-hand graph, which in the yellow you can see trials in myeloma, 1985 to 1998, predominantly younger patients with myeloma. Over that 13-year period of trials, there was no improvement in outcome for patients with myeloma until the first trial published over 20 years ago now, in 1996 from the French, which demonstrates that patients who received an autologous stem cell transplant were living longer than patients who didn't. And that's the graph with the curve on the top being those who received the transplant. If we then look at what's happened more recently, and this is just some data about younger patients, that is those that have a transplant, there's really been a lot of improvement compared to 20 years ago. The median survival for patients that have standard risk myeloma, so this is patients who don't have problems in their genetics that predict a higher risk of relapse. The median survival in these registry and recent trials is over 12 years. And that's a marked advance on where we were 20 years ago when it was less than 5 years. In the top graph, top table, you can see some data from the Canadian registry, similar to our own registry, where they're looking at outcomes for myeloma. And the reason I've put that in is that outcomes in trials are always better than in the real world, because trials pick fit patients who don't have marked health problems, whereas registries like the Canadian one are a better reflection of what's happening in the real community. And their results are maybe not quite as good as the trials, but certainly very impressive. And if you can see in the bottom right-hand corner, this is the most recent data from a trial that we participated in Australia as well as internationally, which was a trial looking at the standard treatment we have now, VRD induction, followed by transplant, followed by rituximab maintenance. Patients have amazing outcomes. This is overall survival out to about 4 years, and still at that point, more than 90% of patients are alive. So there's definite progress being made. I'm just going to talk briefly about the cause of myeloma to lead into the next topic of therapies. We don't know for sure what causes myeloma. It probably is genuinely bad luck, but it's not caused by infections. As far as we know at the moment, it's not preventable. Nearly all cases are preceded by a precancerous phase called MGUS, and that's been shown in studies of US veterans who have blood tests taken often decades before they develop myeloma, and you can go back and see this precancerous phase. But we need a better understanding of disease biology from research to work out what's causing myeloma. There may be a role of what we call the bone marrow environment, the other cells in the bone marrow that surround the myeloma cells. How do they impact the ability of a normal plasma cell to become a cancerous one? And there's certainly a link between the immune system and cancer across all types of different cancers. So if we look at the immune system, we know it's important in all sorts of cancers, and it's certainly important in myeloma. But obviously, for as myeloma is progressing, it's learned to evade the immune system. So would it be possible to retrain the immune system to fight myeloma and overcome the sneaky things that the myeloma cancer cells have done to make themselves resistant to the immune system? Your immune system's got a number of different cells in it. It's got B lymphocytes. These are the cells that mature to go on to produce antibody. There are T cells, which do much of the direct killing of different cells, including cancer cells, and they've got NK or natural killer cells. So there's all these different cells in the immune system that are there to do the work. There are myeloma cells, which can have a coding which could be recognized potentially by drugs if it's different from what's on normal cells. And then we can use that immune system to try and direct one of these cells, the B cells, the T cells, the NK cells, other immune cells, to try and target something on the myeloma cells so that there's less killing of other cells in the body, which is one of the disadvantages of chemotherapy. So in terms of myeloma immune therapy, we've got a number of different processes which have been developed. There have been vaccines developed to try and target your immune system to attack the cancer cell, just like we use vaccines to target them to treat infections. There's CAR T cells, which we'll hear a little bit more about. Monoclonal antibodies, such as daratumumab, are a type of immune therapy. We've got bispecific antibodies and T-cell engagers, which force various immune cells to attack the myeloma cells. We've got antibody drug conjugates, where an antibody selectively delivers some sort of toxin direct to the myeloma cell and not to the rest of the body. And then we've got checkpoint inhibitors, which are widely used in other types of cancer, but haven't yet proven to be useful in myeloma. The thing with trying to target the immune system to a cancer cell, though, is you need a target on the cancer cell. And in the case of myeloma cells, we have got this target called BCMA, or B-cell maturation antigen. It's expressed on all plasma cells, including myeloma cells, but little is expressed on any other tissues in the body, and therefore it's a good target. Its overexpression is associated with progression of myeloma, and therefore is very useful to direct the immune cells to bind onto this BCMA to facilitate the killing of the myeloma cells. It's not the only target at the moment. There's a number of others. There's targets called GPR, C5d, FCH5R, SLAMF7. The actual kappa and lambda light chains can be targets as well. So there's multiple targets that have been developed or have been discovered, but used to target myeloma therapy. So in terms of BCMA targeted immunotherapies, there are the number that are useful. There's antibody drug conjugates, and this is belantamab. There's CAR T cells, of which there's 2 products that have been developed and commercialized at the moment, and they attack the BCMA target on the surface of myeloma cells. And there's bispecific antibodies, again, 2 of which have been commercialized, teclistamab and elranatamab, both of which bring your killer T cells into close proximity with the myeloma cells. So I'm going to talk a little bit about each of these. So firstly, what is CAR T cells? So the CAR T stands for chimeric antigen receptor T cell therapy. So the chimeric antigen, bringing together bits of different proteins to force the T cell to attack directly the myeloma cell. So it's an immune therapy that changes some of the body's T cells to improve their ability to fight cancer. Their production is actually a complicated process. First, the T cells have to be collected from the blood. a little bit like stem cells are collected, but in this case, we're trying to collect the lymphocytes in particular. Then those lymphocytes have to be manufactured in a laboratory. That is, the target has to be introduced into them so that they're focused on attacking myeloma cells. You have to have some sort of therapy to control the myeloma while this manufacturing process is being undertaken. You then have to have some sort of chemotherapy to make sure that the body's immune system doesn't immediately reject the T cells, the CAR T cells that are being made, and then they have to be reinfused back into the body. How do they work? Well, they're a type of white cell, the T cell, that normally is responsible for attacking cancer cells or infectious cells. And their function is to identify abnormal changes in a cell and then to destroy it. In terms of CAR T cells, they've been specifically re-engineered in the laboratory to change a property on their surface. So they've added a gene so that their surface receptor is recognizing something on the myeloma cells so it can then attack them. Now, there'll be a bit more detail about CAR T cells in some talks to come. But I'm just going to mention there's been a number of trials conducted and reported looking at CAR T cells, and these are just some of them. But I just want to briefly mention one trial that's being completed recently, the CARTITUDE-4 study. So this trial was only recently published at the end of last year, where it was comparison of the CAR T cell against one of our most active combination, which is pomalidomide, daratumumab, and dexamethasone. There was another choice, but mostly patients received this therapy. Patients either received the CAR T called cilta-cel or that therapy that had relapsed myeloma. Their myeloma had to have progressed while taking lenalidomide, which is very relevant to our context because most patients receive lenalidomide maintenance as part of their first therapy. And then they had to have had 1 to 3 prior lines of therapy before going onto this trial. So if you had more advanced 4 or 5 lines of treatment, you couldn't get onto this trial. And this trial was important because it was our gold standard, that is a randomized controlled trial where half of patients get one therapy and half get the other. So this is where we really see how good a treatment is. And these were the results. On the left panel, these are the response rates. So how many patients get a very deep response? This is a complete response. So using our common blood and urine tests, this is where we can't detect the myeloma anymore. And you can see markedly improved in the CAR T group, which is the orange bar graph compared to the right, the blue one. And this translated to improved control of the myeloma. So the curve on the bottom right, called progression-free survival, is a curve of how well the myeloma is controlled. And you can see 18 months down the track, markedly more patients have not had their myeloma relapse in the CAR T group in orange compared to in the blue group. So this was definite proof of the effectiveness of CAR T compared to the best available therapy, even better than what we can get in Australia on the PBS. So proof that the therapy is definitely efficacious. But you may also have noted that patients continue to relapse, even those who are getting CAR T. So why do they not always work? It could be that the myeloma is too aggressive and some patients progress in that time even before they can get to having the CAR Ts. In some patients, the CAR T cells just seem to be less effective. Or it may be that they don't persist long enough in the body to exert ongoing effect. It could be that the myeloma cells learn to not express the target that is being attacked by the CAR T cells and they evade the immune system. Or it could be that there are other mechanisms that the cancer cells develop to evade killing by the CAR T cells. And all these mechanisms are under investigation to try and define ways to stop the myeloma cells being able to evade the CAR T cells. In the future, they'll be looking at trying to get the T cells earlier, to see immune system and the T cells work better before someone's had many lines of chemotherapy. There might be the opportunity to develop CAR T cells from other donors, so-called allogeneic CAR Ts or allogeneic NK cell therapy, so-called off-the-shelf treatments, which can be available straight away. Or maybe there's the use of other targets besides BCMA. There's also attempts to optimize the manufacturing process so it can be sped up to get CAR T cells that are more efficacious or have less side effects. There are, though, alternatives to CAR T cells. We're mostly familiar with the plain antibodies such as daratumumab, which binds to the myeloma cell, and then one of the immune cells latches onto that antibody. We've got drug conjugates, which I'll come to, but I want to focus more on bispecific antibodies. These are antibodies that have been manufactured, so one half of the antibody binds to the myeloma cell, and the other half binds to your T cell, thus forcing your cancer-killing T cell right next to the myeloma cell and forcing direct killing of the myeloma cells. So these bispecific antibodies are what we call an off-the-shelf product. They're like a drug. They can be available at any time to start treatment and don't rely on the time needed to manufacture CAR T cells. And there can be so-called bispecific antibodies or bispecific T cell engagers, but they both have the same principle that can be seen in the picture on the right. They bring your T cell into close proximity to your myeloma cell to force myeloma cell killing. There's a number of these types of drugs in early phase clinical trial and in some in advanced phase clinical trial. And two of them have achieved registration in the US, but not yet in Australia. They've got reasonable safety profiles. They do have cytokine release syndrome, although that's manageable in most patients and a little easier to manage than in CAR T cells. But they do have problems with increased rates of infection down the track, and this is an area that particularly needs to be improved. Most are intravenous or subcutaneous injections weekly and then either fortnightly or monthly, so they are a therapy that gets easier as the treatment progresses. Most of them at the moment, because they're used in later lines of therapy, are ongoing until disease progression. But the responses are very impressive, 62 to 83%, compared to 30% or less with most traditional therapies. Here's a slide with a lot of detail in it, but does show there's a number of trials that have reported out with different bispecific antibodies in myeloma. There's actually probably more than 30 trials underway in various stages of conduct. These are the trials that have been reported in the relapsed setting using drugs against BCMA, teclistamab, or against one of the other targets, talquetamab. There's elranatamab, which has also been used against BCMA. You can see all of them have been used in different populations, typically patients that have received a lot of other drugs. And their response rate has been so promising that just like CAR T cells that have been moved to assess earlier lines of therapy, indeed being used upfront as initial therapy, to see if outcomes with the current treatments can be improved by bringing more efficacious therapy upfront. So in summary, with these bispecific antibodies, there are several that exist in advanced stage of clinical trial and also in the stage of becoming available around the world. They target different targets on the myeloma cell. All of them force your T cell to directly attack the myeloma cell. The maximum response and duration of response is yet to be fully determined. We need some more follow-up on the trials. It's possible that future will be combining them with other agents to maximize their tumor killing, and more work needs to be done to improve their safety profile, particularly in regard to reducing infectious complications. So we now have CAR T cells and bispecific antibodies, which both use the immune system to directly kill the myeloma cell. And they've both got pros and cons should they become available. There's no doubt that the bispecifics that are available straight away for patients, they're a product that can just be sitting on the shelf, whereas CAR Ts do have to undergo manufacture in specialized centers. They probably will be a limited number of centers where CAR Ts can be given, at least initially. Hospitalization is required, at least in Australia, in most centres at the moment for CAR T cells, whereas there is less hospitalization required for the bispecific antibodies. A great advantage of CAR T cells is a single one-off administration with no ongoing therapy, whereas bispecifics are ongoing treatments required, even though the schedule may become less intensive. There are toxicities of both approaches, which you'll hear about in a subsequent talk, and they're certainly both expensive. We don't know how they'll be priced exactly in Australia yet, but rest assured they won't be cheap. I've mentioned a couple of final points on other treatments. Belantamab is a drug where the BCMA antigen is targeted on myeloma cells, but a toxin called MMAF is attached. This is a treatment that looks effective, but it's got a unique toxicity in the eye, and its best way of combining with other drugs and positioning in myeloma has yet to be fully worked out yet. A recent trial looked at belantamab, didn't meet its endpoint of being more effective than pomalidomide, and so more development needs to occur. The next generation of the thalidomide family, the so-called CELMoDs, is in development. Iberdomide and mezigdomide, these are very powerful agents, oral agents, so they're attractive and will probably be used mostly in combination or as maintenance therapy. I won't go through the data on this trial comparing the different efficacies, but clearly the CAR T cells and the bispecific antibodies are very effective compared to some of these other agents. In conclusion, the future of myeloma is bright. There's significant progress made from decades of research to discovering new therapies and translating them into trials and availability for patients. There are multiply active immunotherapies at the moment, which appear very promising. And the current issue is to, one, get them available, but to work out how to optimize the timing, sequencing, and management of side effects. Just to give you a pointer of how far we've come, 40 years ago to 30 years ago, all we had was melphalan and prednisone. Complete remissions, 2% at best, 1 year survival, 20 to 30 of patients always died in the first year. We couldn't make an impact on that. Transplant improved things further. If we come down to the recent trial of VRD and daratumumab, 80% complete response rates. But if we look at CAR T cells, we're getting similar complete response rates to our upfront therapy, even though this is in patients who have received 6 prior treatments. They really are extremely effective therapy in terms of getting myeloma under control. This is a study that was published a few years ago looking at relative survival. So this is the survival of someone with myeloma compared to the aged and gender-matched population. So you can see over the last few decades, we've come an awful long way from a relative survival of around 25% to over 50% with the development of therapies. We've still got a way to go to get the relative survival to 100%. That is, to either make myeloma truly a chronic disease or to cure it. But progress is being made. That's all I've got to say. I'm happy to take questions. It's an exciting and optimistic time for myeloma. Thank you.
Thanks so much, Dr. Malay. We do have a couple of questions that have come through the line. We have one from Babsy, who asks, how far away are we from Dara as first line, and will we get quad therapy in Australia?
So we don't know the answer to that. I mean, daratumumab in combination with Revlimid and dexamethasone has been submitted twice to our regulatory authority, the PBAC. It's been accepted as a safe and efficacious combination by the TGA. but the issue is primarily around price. So I guess it's being submitted again, and look, we really hope that it'll become available. It really is a very effective but very tolerable therapy, and this is very important for older patients with myeloma. In terms of quadruplets, I guess the company hasn't yet looked at making submissions to the government for upfront addition of daratumumab. It has been shown to be effective in younger patients treated with VRD and transplant. That data has just been published. So we'll wait and see. I'm sure that'll be in the company's plans, but I don't think that will be this year.
Thank you. A question's come through from Bill, who asked, do we know what causes myeloma?
No, we don't. I mentioned bad luck, and that probably is the case. There's a small number of situations where we know that myeloma is caused by. One of those is high levels of radiation. So people in Hiroshima, Nagasaki, Chernobyl, they develop myeloma at greater rates. In fact, one of the people who worked on the atomic bomb in the US developed myeloma 20 or 30 years later. So there's no doubt that high levels of radiation are important. But apart from that, there's a lot of uncertainty as to the exact causation of myeloma.
Thank you. A question about, is transplant still the best therapy available in Australia?
So for younger patients who can tolerate the procedure, it still is. And as every new therapy has come along, there's been attempts to replace transplantation. When lenalidomide came along, a trial was done to compare, a regimen with lenalidomide versus transplant or not, and outcomes were not improved. It remained superior in the transplant arm. Velcade came along, the similar trial was done. Velcade and Revlimid combined came along and a similar trial was done. There is a trial being conducted in Australia and internationally at the moment, which will directly compare CAR T cells with autologous stem cell transplant. And we'll see if that shows that a therapy can beat transplantation in terms of control of myeloma. But to date, it still seems to be a highly effective therapy, more effective than not having a transplant. That only applies, though, if you're fit enough to have a transplant, and that's a complicated assessment.
That leads nicely into probably the next question as well, which is, if you haven't had a transplant, would you still be eligible for CAR T?
So at the moment, there's nothing in the wording about CAR T cells about having had to have had a transplant or not. It's all about lines of therapy, of which transplant is one. I think we have to try and get it available in Australia first. And again, it's up before our bodies that assess the availability of funded therapies in Australia. And we keep our fingers crossed that there'll be a positive outcome for that submission. But it comes down to the price of these therapies rather than their effectiveness, which seems to have been accepted. It's really, can Australia afford that therapy?
Absolutely. And I think, as you alluded to, then working out the sequencing, isn't it, where we best fit in these lines of therapy. Questions still coming through. Thank you, everybody. We've got one here. My hematologist has advised a BITE trial as I have relapsed for the third time. What things should I consider?
So I think when you get to your third relapse, there's no doubt that the response rates to bispecifics look superior to any other option that we have. The things I'd consider is it a trial where there's a choice between two therapies, the BITE therapy and something else? And what is that something else? Is that suitable? It's highly likely on most BITE studies in Australia at the moment that the control arm is better than what we can get on the PBS at the moment. So you should look at that carefully. I think, though, if you look at every therapy that we have available in Australia at the moment for myeloma, it's because a patient before you has participated in a trial and proven that that therapy is effective. And on the basis of those trial results, it then becomes available for everyone. So I would say, consider the data in the trial carefully. Consider what your doctor's saying carefully. Speak with your family and your GP. But I think generally speaking, the care delivered on a trial is high quality, and you will be helping availability of treatments for patients in the future.
Great, thank you. And what looks like our final one, there has been a recent report about secondary T-cell malignancies after CAR-T. Can you offer any further information?
Yeah, so there was what they call a black box warning in the US that's been put out on all CAR-T products. And this relates to the occurrence of a greater than expected frequency of T-cell lymphomas. So T-cell lymphomas are a cancer of the T cells, and part of the manufacture of CAR-Ts is that those T cells are manipulated. So at the moment, they've had more cases than expected, and some of them, they suspect that the CAR-T manufacturing process might have led to the development of those lymphomas, but the data is a little bit too early to be sure about that. So it's been listed as a warning. All centres that administer CAR-Ts have to track long-term for the development of these types of cancers that may be related to the CAR-T process. At the moment, they seem extremely uncommon. So if you like, the benefit of the CAR-T in preventing progression of the myeloma, in fact, getting it into remission, far exceeds what seems to be a very uncommon event at the moment. But I think all of the side effects of CAR-Ts and this report of possible T-cell lymphomas is but one, need to be investigated, and there's a lot of research being done to try and reduce risk of these things. So a lot more to follow in this story about T-cell lymphomas, early days, and at least for the moment, a very uncommon side effect.
Right. Okay. Thank you so much, Dr. Malay, for your time and your expertise. A reminder, if you do have questions, you can send them via our nurses inbox, and we can reach out to Dr. Malay after or, assist in answering those questions. Thank you so much again. I'm going to
Thanks very much, Kath.
Thank you. Handing over to my colleague, one of our specialist myeloma nurses in Victoria, Danielle Hovey, to introduce our next speaker. Thank you.
Thanks, Kath. I have the pleasure of introducing our next speaker, Dr. Wojt Janowski. Wojt sees patients with thrombotic and other general hematological problems, as well as those with leukemias, lymphomas, myeloma, myeloproliferative neoplasms, and myelodysplasia. His main interests are the diagnosis and treatment of hematological malignancies, and he is the clinical lead for trials in multiple myeloma and CLL at the Calvary Mater. He works on protocol development and the rapid translation of cancer research into clinical setting. He's involved in every level of medical training, from medical students to hematology advanced trainees, coordinates a hematology training program for general physician trainees, and has spoken at various GP events. After completing his physician and hematology training in Melbourne, Wojt joined the hematology team at the Calvary Mater in 2014. He started in private practice in 2015 and has relocated to the Hunter Cancer Centre at Gateshead to be able to provide a more comprehensive consultation and cancer treatment service. Wojt's presentation is called Managing Expectation of Emerging Therapies. Thanks, Wojt.
Thanks very much, Danielle, and what a talk to follow, Peter. Thank you very much for that. Even I learned something from that, so that was great. Thank you. Look, as Danielle said, I'm a hematologist, both in public and private practice in a lovely city called Newcastle. If you don't know it, it is neither new nor has any castles in it. But it is a great place to live, and you'll never shift me from that at all. As time has gone on, and while I certainly used to treat all of those things and continue to do, most of my work at the moment is now on treating people with myeloma, and particularly on trying to attract more clinical trials to regional centres, particularly our own. And we've been very fortunate to have excellent industry partners, as well as support from our colleagues around Australia, and we're the first centre outside of a capital city to administer CAR T cells, which was in the context of a clinical trial about 8 weeks ago. So a huge amount of effort by our staff. So that was a great achievement, and so I'm very excited about what this new era of medications brings to patients with myeloma. I think Peter's just talked about some extremely exciting therapies, and I 100% agree with him that they are all very, very exciting. And we really appreciate being able to give them to patients in the context of our clinical trials. So when Myeloma Australia asked me to give this talk, I can't say I was overly enthusiastic, because I really feel like I was asked to rain a little bit on the parade. But it's not really that. I came around to the idea and realized that it's all very well reading and hearing about all of these extremely exciting therapies, but if you can't get access to them in a timely fashion, then is it just, is it false hope, or what can you do? And so I think when we're reading about all of these things which are available in clinical trials and are available overseas, we also have to be realistic about when we are going to be able to get them into routine clinical practice in Australia. We work extremely hard to get them here on clinical trials, but only a tiny minority of our patients will actually benefit from clinical trials themselves, because they are, by their very nature, small and often not applicable to many people, as we'll cover later on. In the end, when thinking about these things, I'm always focused on the fact that our goal is ultimately cure of myeloma and what that means. And I am extremely excited by the progress that has been made, even in the last 15 years, in the treatment of myeloma. And Peter showed that lovely slide of how with every generation of myeloma medications, people with myeloma live longer, and that's fantastic. But ultimately, we have to remember that, I don't have to tell this audience, that ultimately what we want when it comes to a cure is a treatment you can have for a limited duration of time. That basically means you are done with this disease, that life is back to normal, that it hasn't caused you lots of problems as transplants can do and other treatments can do, and your life expectancy is now what it would have been without myeloma. And so when you look at where we've come from, you can be really, really happy. And when you look at where we still need to get, you can tell that there's still a great deal of work to do. And clinical research trials of getting these medications early is part of that process. So what I'm going to talk about today is, first of all, what a long road it is from conducting a clinical trial to actually being able to prescribe a medication on the PBS, and for someone with myeloma to get it for $35 or $39 a dose or prescription, and how complicated that process really is. I'm going to talk a little bit on why often what we may expect from a medication doesn't actually match up in what we consider to be the real world, which is PBS prescribing. And then, since I will be talking and encouraging the participation in clinical trials, talk about the maze of accessing those and what that means to you as a patient in a clinical trial, what you might need to know in general terms. So if we think a little bit about how a medication gets from a good idea or from a laboratory to the PBS, there are very many steps on this road. And in fact, before we even get to that phase 1 first in human study, there's a lot of research being done predominantly at universities and small biotech companies, where they're taking good ideas or things they know about the basic biology of myeloma, and testing it out on cell lines and testing it out on rats and trying to find new medications and new chemicals that may actually be helpful. That phase can take a very, very long time. The first concept that something like a CAR T cell may be able to work was in sort of 1987. That was when people first conceptualized that a CAR T cell may work. The first time one was actually given to a human being was 15, 16 years later. The first registration of any CAR T cell for any disease It took another decade from there. So it's a very, very long process. But basically what happens when it comes to actually people receiving these medications is that the company, first of all, has to do studies which are considered higher risk on very small numbers of people. And these are the first human beings to ever receive these medications. Someone's got to be number one, right? And while you try and predict as best as you can what the side effects might be, and as best as you can what the correct dose might be, in the end, somebody has to be first. And so this is a small number of people, very carefully controlled studies, generally done in very large centers. We need a lot of support and backup to be able to do the tests properly. So very hard to find these outside of capital cities, although we do nudge into these a little bit in some centers. And really, these are studies for people who really have no better option left to treat their myeloma. So all your good options have been exhausted, and you're now trying to find something that may well be of help. And a lot of medications actually fall over at this stage. We know that a lot of medications won't work, and most of us will never hear about them in the first place. But say it does, say it looks promising, it doesn't cause any terrible side effects. Well, the company's then is going to do some larger studies. And so instead of 10 or 20 patients in the phase 1 studies, we'll probably take this up to about 50 patients. And here they're trying to better define what they know about the medication. So these studies, we've got some idea about how to dose the medication, but we're really trying to optimize the doses, trying to find the absolute best dose you can use, as well as trying to combine it with other myeloma treatments and seeing whether you have any sign that there's actually a synergy, that 1 + 1 is greater than 2, and making sure that there isn't any surprising side effect when you add them together. And so that's really important. And so at this point in time, another 50-odd people have received the medication. If it looks really promising at that point in time, in other words, it looks safe, it looks well tolerated, and there's an indication that the medication is going to be effective as well, what the company then needs to do is they need to prove that it actually works compared to what we have already. And those are the phase 3 studies. So generally speaking, this is the first time that you'll see a clinical study where there's any randomization involved. And so these are studies where a person will agree to participate in the study, not knowing whether they will be receiving the standard treatment or whether they will be receiving the newer experimental treatment. And these are really important because not every treatment out there is actually going to prove to be a good idea. Some medications are going to have side effects that are going to make them less attractive than what we have already. And there's no point having a treatment that is extremely potent and able to kill myeloma cells if it makes people's lives miserable and the side effects stop them from being able to take it. And there's also no point having an extremely modern, expensive medication that doesn't work any better than what we already have. And so this is where those phase 3 studies are crucially important. Because in Australia, as you'll see in a little bit, we tend to be a little bit behind in registering new medications. And because these studies take a really long time to run, even with the control arm, even the one that's the standard treatment, in some of these international studies is actually better than what we can give patients in Australia at the moment. So somebody asked the question about daratumumab in the front line. And if you look at some of the phase 3 studies we have at the moment that are looking at medications like teclistamab and similar medications being added to the front line, the comparison there is against daratumumab, lenalidomide, and dexamethasone. So that's a scenario in which you might not get the newest stuff, but you'll still get something better than PBS. So those are really attractive studies to consider. Anyway, once all of that research has been done is where you actually get to the point of talking to the regulatory authorities. And this is very different around the world. So in the United States and often in Europe, companies can actually approach the FDA at the conclusion of their phase 2 studies. Because the FDA, which is the American equivalent of our TGA, is only interested in, is this medication safe and does it do what it says on the tin? Does it actually treat the disease in question? And if it does, the company then gets approval to market the medication, to sell it in the United States. So that's always the first goal for the pharmaceutical company. That is the market in the world that pays the most for medications of anywhere else on the planet. And that's where they stand to make most of their money. So that's what they focus on. Our TGA is similar to the FDA in that they will accept an early application on the basis of those phase 2 studies. And what that allows the company to do is to sell the medication to patients in Australia and to talk to doctors about it in Australia. But it doesn't have anything to do with government funding. It doesn't have anything to do with the PBS saying, yes, we will pay for this medication. And so really up until this point in time, our regulatory process in Australia is very similar to what you would see overseas. The challenge then is the PBS. And the PBS in Australia is charged with managing the pharmaceutical budget. And this is a very difficult job because myeloma is one of many diseases. And so what they are doing is they are looking at all of the clinical trial data that the company has submitted to them. And they're trying to quantify the benefit to the individual patient. They try to put an effect number in terms of how many patients do we need to treat in order for one of them to have, say, a year of extra life. And then they look at that benefit and they look at what the company has asked the PBS to pay for that medication. And they say, well, what that means is in order to pay for this treatment, if we pay for this treatment, we will have to spend, say, $50,000 for an extra year of survival on average per patient. And they don't have a firm number. They don't have an absolute line in the sand that they require that treatment to meet, because it does depend a little bit on the disease group. It depends a little bit on how many other treatment options are available for that disease. And it does depend on how good the treatment is. But they do ultimately have to come to a decision. And the reality is, it's a bit like buying any high-cost asset, be it a house, or a car, or anything else. The first price discussed is rarely what actually gets approved. And it is extremely unusual for the PBS to approve anything on the first attempt, especially with high-cost cancer medications. There are a number of reasons for this, and not all of them are the government's fault. When the pharmaceutical companies put the application into the PBS, they're also very mindful of what they're selling the medication for around the world. And as I mentioned, the United States, which is always first past the post when it comes to medications, pays more for their medications than anybody else on the planet. And they set the global price for the medication. And everything else after that to reduce the price is a negotiation. And so this process can take several years. We are a small market, we're 26 million people compared to, say, the United States of 360 million people. Getting the medications on our market is a priority for the pharmaceutical companies, but it's not priority number one for them. And so we have to be mindful that we are operating in this sort of environment. This whole process can actually take several years to run its course, and it can be extremely frustrating to watch because as clinicians, we want to use these medications as soon as we see the clinical trial data published. As patients, you want to have access to these medications as soon as the clinical trial data is published. But it might be a 3 or 4 or a longer wait even before we can use the medication. And then, for example, like daratumumab, we may be severely restricted in how we use it. And so this can take an extremely long time. Now, pharmaceutical companies can choose to make the medication available early in Australia. They can do that in whatever method they want. So once they have TGA approval, they can then go out into the community and say, we have this new medication for myeloma. It's going to cost you this much. If you want to buy it, you can buy it and you can have it. But this is how much it costs you. Some of them can choose to do things like offset the cost of that medication. So for example, they might say you can buy 4 doses of this medication and we'll give you 4 doses. So that offsets the price. In other words, it reduces the price of the medication to you, but it means they don't have to reduce the sticker price. And very occasionally, although it seems to be becoming less common, at least for a period of time, they will make the medication available completely free of charge on what's called a compassionate basis. And you can discuss the morality of it, but there is no rule and there is no law that says they have to do that. So when they do that, that's of their own accord. I don't really want to get into that too much because it's a complicated discussion. So that's what the process that has to work for things like teclistamab, etc., for all of those other molecules, but it's slightly different than what happens with CAR T cells. Because, as Peter pointed out, CAR T cells are a product that's manufactured from living cells, they're not actually considered a medication in Australia. They're considered a cell therapy and are regulated as if they were a medical procedure. And so they are regulated by the same authority that looks at, for example, will Medicare pay for this new blood test? Or will Medicare pay for this new surgical procedure? It's the same process to look at CAR T cells. And so the PBAC don't actually have a role to play when it comes to approving the funding for CAR T cells. But the basic principles they follow are the same. Although as an outsider looking in, and I've not had anything to do with the negotiations with MSAC, but as an outsider looking in, it does look like they at least have a little bit more flexibility and room to move because their criteria are not quite as rigid as those of the PBS. On the other hand, it's also a more complicated mechanism because the way CAR T cell treatments are funded in Australia, the federal government has to share the costs of the CAR T cells and the treatment with the state governments. With PBS, the federal government picks up the entire cost. So as you can imagine, that's a more complicated discussion because as we know, state-federal discussions about money are always a little bit more complicated. And so, this is all extremely frustrating. It's frustrating for us as doctors standing in front of these medicine cabinets knowing we can't actually use these treatments yet. And it's frustrating for you as well. We all live in a global community now. And I think it's fair enough that you can ask why treatments that are proven in clinical trials that are in use in other countries are not available in Australia. And I would encourage you to advocate as a community, and I think that Myeloma Australia has been phenomenal in driving this, and particularly with the CAR T cells and the application to MSAC. And I think the myeloma community has been phenomenal in writing to the PBS and writing to MSAC and telling them what it means to them to have these treatments on the PBS and on Medicare. I think that's crucially important. I think I'd write to your MP. I would write to everyone you know of any influence and talk about this as much as you can, because I think it's a big problem. The government recognizes it's a problem, and they are working through a process at the moment where they're reviewing how they fund medications. But all of these reviews take a long time, and it's still a work in progress. The argument from their side is, look, they do have a limited budget, and I'll throw some of the numbers around a little bit later just for interest. They do have a limited budget, and they have a lot of competing health priorities, and not all of the information or not all the clinical trials that pharmaceutical companies produce is absolutely correct. And there are many ways in which you can adjust the design and interpretation of a clinical study to make a medication look a lot better than it may actually be. And so we can't have a system where we rely on the pharmaceutical industry to self-regulate the quality of the information they produce. I think the pharmaceutical industry is extremely important. We wouldn't have the medications we have without them, considering the cost of developing medications. But there needs to be a balanced system. I would argue right now we don't have that balance correct. And I would argue that for a country as rich as Australia, we should have earlier access to these medications. And it's extremely frustrating for everyone involved. So I'm going to switch tack a little bit. And as I mentioned before, sometimes our expectations of these medications may not match the reality. And what I mean by this is that either the treatment is not quite as good as we thought it was, and we get told that this medication is great, this monoclonal antibody or this bispecific antibody has this amazing effect and this great response rate. And then we actually give it on the PBS, and we give it to people on the PBS, and we look back and say, you know what, it's not actually as good as those initial studies suggested. And this is a really important thing to understand, particularly if you're considering buying your own medications. I think you've got to have a very clear idea of, if you're going to be spending tens of thousands of dollars of your own money on medications, you've got to have a very good idea of what that treatment may actually offer you, not just in terms of benefit, but also in terms of side effects. And so, I always, you'll find this placement of the real world in quotation marks in a lot of scientific literature. And I find it very amusing that we are talking about the real world in quotes as if it's some sort of strange entity. But it also, what it reflects is that really the world of clinical studies is a little bit artificial, because patients in clinical studies compared to the patients that come through our clinics on average are younger. They have fewer health problems in general. They often have slower moving myeloma, as you'll see when we talk about what it takes to participate in a clinical study. The time from saying, yes, I'm interested in the study to actually receiving treatment on it can be 3 or 4 weeks. And so if the myeloma is moving extremely aggressively, you may not actually get that treatment. So people with the worst sort of myeloma, that very aggressive disease that's just day by day growing further and further, aren't well represented on clinical studies because they just don't have time to enroll onto them. Because clinical studies are generally done in larger hospitals and more affluent countries, you're looking at a different population in general, and people with better access to health resources. If you look at, the sort of people that get onto clinical studies at these super, at these ultra premium cancer centers in the United States, they're not the same as the people who come from regional Australia. They don't have the same access to resources. And also on a clinical study, the patients are followed extremely closely. And if you've ever participated in a clinical study, you know what all of the extra requirements of being on a study, and you know how closely the trial team watches you compared to routine clinical practice. And that's probably, a reason why people will be able to maintain the treatment for longer on a study than off it. And so when you're looking at a new medication and thinking, should I give it to the patient in front of me? These are the sorts of questions I'm asking about that patient, or these are the sorts of questions you could ask yourself about any new treatment, be it on the PBS or otherwise, is, did the people in the study used to register this medication, did they actually look like me? Were they similar people to me? Were they of a similar age? Because there's no point if you're 75 or 78 or 85 looking at treatments that were the best thing ever in 40-year-olds who could deal with a lot more side effects. We know that from every aspect of medicine. Were they as sick as I am, or were they sicker? What treatments for myeloma did they have? And does that influence whether this treatment will work for it? And after considering all of that, are the expectations I have about this treatment and what it can do for me realistic? Is this treatment and all of its side effects that I am likely, because there's no such thing as a treatment without side effects, are all of these treatments, is it worth participating in? Is it worth taking this medication? Is it worth paying for this medication? And it's not just about the medication itself sometimes. If you look at the BCMA targeting bispecific antibodies, and there's a few of these around, teclistamab is registered by the TGA in Australia but not funded. There are a few other ones in clinical studies. And if you look at all of those medications, what you can guarantee is that people are going to end up on replacement immunoglobulin, Intragam. And people who have been on Intragam to boost their immune system know that that's either something you have to inject yourself under the skin every week, or it's something you're receiving in a hospital every 4 weeks. And that's an ongoing thing from what we can see. And that's not often talked about as a side effect, but from your perspective as a patient, it means you have to be somewhere every 4 weeks because you need, those top-ups of antibodies. These are all things that need to go into consideration when looking at some of these new treatments. So think about questions like, how often do I need to take the treatment and where can I have it? Do I have to come to this hospital to have it? Could I have it at home? Could I give it myself? And some of these things are possible, for example, having treatment at home, but the availability of it is very lumpy around Australia and very, very patchy. How long does the treatment go for? Most myeloma treatments these days are actually indefinite. In other words, you start on the treatment and you carry it on until it stops working, or until the side effects cause you to stop, or until you finally decide, I've actually had enough. We know carfilzomib is on the PBS. Carfilzomib is a very potent medication and very, very useful. But the original treatment schedule with carfilzomib was twice a week, intravenously, 3 weeks out of 4. That's a huge commitment. And all of these other questions. So, ask your hematologist, sure, this is my 5th treatment for myeloma. What are the realistic chances this is actually going to work for me? And if it does work, how long will it work for? And I think that's a really important question that you go into these, any treatments and any trials, understanding that information for yourself. The other reason why reality and expectations may not meet is because of this problem of accessing the treatments anyway. We've been talking about CAR T cells in these patient seminars for at least a couple of years now. And so where are we up to in Australia? This is the latest update based on everything that's available publicly. So CAR T cell, cilta-cel, is the only registered CAR T cell in Australia for myeloma, cilta-captogene autoleucel or cilta-cel. The TGA approved it sometime last year. And so that means theoretically, you could buy them. I'll show you why that may not be such a great idea in a minute. But even practically speaking, at the moment, that's not possible. And the discussions that the company is currently having with federal and state governments and MSAC is as a treatment beyond 4 lines of therapy. To show you how long these applications and negotiations take, the first application, which probably took about 6 months to put together, was considered by MSAC at their meeting in June 2022, almost 2 years ago, at which point they basically said, the data is immature, we don't know whether this is really as good as you say it is, and oh, by the way, it is far too expensive. The company immediately started working on the next submission. And the next submission was considered by MSAC November last year. And so that is a very long time between, sitting down, between, meetings at the table to actually sit down and talk about what's going on and will you pay for it. And a huge amount of progress was made at the second application from what I can see. And I'll show you some quotations in a second. But still, the government essentially came back and said, it's still too expensive. So if you look at the publicly available minutes, I know most people have more of a life than I do, so I will quickly highlight the bits that may be interesting, is that on the government side, they acknowledge that people who are at this late line of myeloma treatment don't have a lot of options, and that cilta-cel, that CAR T cells absolutely work, and they work better compared to the previous. However, they still, if you read further down, it's too expensive. And on the basis of both the MSAC committee and the submissions from the states and territories, they basically said, we can't go there, we can't fund this until CAR T cells are significantly cheaper than what they are today. And in the minutes from all of these things, the company always gets a right of response, which gets published along with the minutes. And I am not implying anything about either side in this debate. I just want to show you where the discussions are up to at the moment. And basically, Janssen, who's the manufacturer that sells CAR T cells, said, look, basically, we've tried really, really hard to sharpen our pencils and get the cost down. We've reduced it by 40% over the 5 years compared to what we asked before. So if it's too expensive at a 40% discount, imagine what the original price is like. But they're officially saying, look, basically, until you guys recognize that this is a really, really good treatment and therefore it's expensive, we're kind of stuck. That doesn't fill me with confidence. But as you can imagine, there is a huge amount of motivation to get these things funded, to get people treated. And a deferral, which is what I pointed out happened at the last meeting, is a little bit different than a rejection. A deferral says, basically, from what I can see, we're almost there, but there's a little bit more work to be done. And hopefully what that means is they can sit down at the table sooner than another 18 months and have further discussions. So fingers firmly crossed. Why is the government being like this? Why is the government being so recalcitrant in funding these amazing treatments? And this is what scares them. So I am using publicly available numbers here, and I'm taking this from a paper published in the US. So while I think the cost of CAR T cells is probably about right if you were buying them yourself, the cost of care in Australia is probably smaller than that, $250,000 to, and that is delivering the CAR T cells. It's all the apheresis, it's all of the treatment, it's everything else. And so if you look at all of that, if you basically, I don't want to spend too much time on this, but basically they're just scared of a cost of $1 million a patient. And if only 10% of people diagnosed with myeloma ever in Australia were diagnosed with it, you're already talking about $250 million a year. And that is, as you can imagine, extremely frightening if you're the PBS, particularly when you consider that the entire PBS budget's about $15 billion. And myeloma is less than 2% of all cancers that they have to fund treatments for. So I don't blame them for having difficulty with the cost. I just really hope we can find a way to get over it. The other questions about the bispecifics and where are we at? And the reality is that while we are using a lot of teclistamab in research studies, there is no phase 3 data yet, and the PBS needs phase 3 data before they will consider the application because they need something to compare it to. So it's not here soon. The current cost, if you choose to purchase it, is approximately $7,000 a dose, and the official prescribing schedule is once a week. I wouldn't ever use it once a week. Most of the studies now don't use it once a week, but that's the official cost and the official line at the moment. And the company says, look, we know it's expensive. We'll at least give you 4 for every 4 that you buy. So at least it kind of halves the price of it. And if you're the right patient, if you fit the sort of the trial criteria, in other words, you're a similar patient to the patients treated on the studies, the chances of it working are about 2 and 3. And so you're spending about $28,000 before you know whether it's working. And then if it is working, you've got to think about how you're going to fund it after that. Pretty frightening stuff. And so it comes to the fact that for a lot of these newer medications, and particularly the stuff coming down the track even now, the only practical way to access these treatments, if your Powerball ticket didn't come up this week, is to participate in a clinical study. And that's something you need to always be thinking about and considering. First of all, you've got to find a clinical study. And just because, you have to recognize that studies that research studies come and go at every hospital. They open and shut depending on what's on the study and what's happening with it around the world. And they're targeted at different patient groups. While imperfect, probably the most useful tool as a general statement, if you want to go looking around, is this website called ClinTrialRefer. It can take a little bit of work to find your way around where the myeloma studies are, but you have to go into hematology first, and then multiple myeloma, and then Australia, and then your state, and then your hospital. And that is a reasonably accurate guide to what most hospitals have available, although it's not 100% accurate. And the other thing is always be asking your clinician when it's time to choose a new treatment. Say, are there any studies nearby? Do you have any studies here? Or are there any studies in a hospital that I could travel to that may be of benefit to me? And I think that's always really important to ask. It's not as straightforward as having the study available to you, as there's a number of different steps that you'd have to go through in between enrolling on a study, or sort of agreeing to be on a study and actually getting treatment on it. And so generally speaking, what we will do at our hospital is, before we even suggest a clinical study to someone, before we even know whether they might be interested, we first of all have a look at the criteria that the study has, and these are called inclusion and exclusion criteria, to see whether the person would qualify for that study. Because I don't want to be telling you about a clinical study that may be available to you if you're never going to qualify for it anyway. What's the point of that discussion? What's the point of raising hopes that there might be something available if it isn't? And so a lot of the time we will look at people and say, no, I'm not going to raise that one because we know their kidneys don't work well enough to qualify, for example. But say the study does look like it'll work for you. You do look like a good candidate, or a potentially good candidate for that study, or you may be interested in it. And I, as a clinician, believe that it's worth your while considering. You will get told about the study, and you will receive a often 20 to 40 page consent form. This consent form, which is much more complicated than it needs to be a lot of the time, outlines everything that participating in the study will involve. It also outlines all of the potential downsides and all of the potential risks of being involved in the study. And that includes all of the medications. So it'll list all the horrible effects of dexamethasone and all the horrible effects of lenalidomide and bortezomib and all of the other things that can happen. And so sometimes it's almost too confronting to read all the potential side effects, when sometimes the reality is the other option is having a lot of those medications on the PBS anyway. But say you read all of that and you ask all the questions you want to ask, and you say, yep, I'm going to do it. I want to be in this study. That's the point at which you sign the consent form and go into a phase of the process called screening. Screening is a process where you have a lot of tests to make sure that everything about you and the myeloma that you have fits for the clinical study, so that you are the kind of person that they are looking for the study. In other words, you've had a certain number of treatments and the myeloma has done this, and you've had these treatments in the past. And also making sure that you don't have any health conditions that make it more dangerous for you to be on the study. And so if a medication can cause neuropathy and you already have a lot of neuropathy, then they won't let you onto that study. If you meet all of those criteria and it is a randomization step, that's sort of when you get enrolled onto the study properly. If there's a randomization to different treatments, that's when that happens. And then finally, you can get onto treatment. And as I mentioned, that can take up to 4 weeks to get that process from consent to treatment finished. I'm pretty confident that's actually my final slide. A couple more. There are potential downsides of being on clinical studies. I think the reality is while we always think that the newer, shinier thing is better, that's not always the case. And sometimes there are unforeseen side effects that people have to be mindful of. The reality is that on a clinical study, the treatment schedule of when you get each medication is much more rigid than it is on routine PBS treatment. So, for example, I've got lots of patients on carfilzomib. If they're on carfilzomib on PBS and everything's going really well, and they say, I'm going overseas for a month, I say, great, away you go. If you're on a clinical study and you're getting treatment and you say, I want to go sailing around the world for 3 months, we have a problem. Because while there is always room to move and negotiation, and it's not that you can't interrupt the treatment, there are limits put in place to what you can do. So, in the first 3 months of a study, you might find it very hard to change the scheduling, whereas maybe, a year down the track, it's a lot easier. And of course, it's very different depending on what's going on and what the study looks like. You will inevitably be having more blood tests. You will often have more scans and generally more tests. There are diaries to fill in. There are quality of life assessments to fill in. And unfortunately, there are more bone marrow biopsies as well. For a lot of myeloma studies. And it's often because you're doing very well and you've achieved a complete remission. And that's when we ask you or complete response. And that's when we ask you for another bone marrow sample to in order to confirm that there's no myeloma left in that bone marrow and do tests like MRD and things like that. And so it is a bigger commitment. In fact, one of the hematologists at Peter Mac said, quite happily says that being on a study can be like a full-time job. I think that's not quite as bad as that. And I have plenty of people who have full-time jobs who participate in studies, but it is a time commitment. And so you need to have that motivation to do that. But the personal upsides for you being on a clinical study is, of course, having access to treatments you might not otherwise have access to. And I always laugh at being on a study sometimes is you have this concierge service from the trial coordinators where you've always got a point of contact. You've always got someone interested in every side effect that may be happening. And you're generally watched by a whole team of people, even if that is remotely. There's a lot of people looking very carefully to make sure that you're getting the treatments as you're meant to be getting them and that the side effects are being noted and managed. And in a way, participating in a clinical trial is how you make that, those expectations perhaps come a little bit closer to reality. Because, if you say, well, how do patients do on clinical trials? Well, that could be you as well. You're on a clinical trial. So that's, I think, everywhere I had to get. And I'm grateful that Peter finished his talk a bit early so I could ramble on a bit longer. But I'm happy to take any questions.
Thanks, Voigt. That was a really interesting presentation, and I'm sure that our community got a lot out of that. So thank you so much. We've probably got time just for one question. So I will email the other questions to Voigt and our team, and we'll respond to those that we don't get to. But one question we had was, how can somebody who lives in a rural or remote location access clinical trials? They are mainly in metropolitan centres. Is there an option to access them from their local treating centre, but be part of a bigger trial from a metropolitan location?
Yeah, I would love for this to be simpler. The reality is a lot of the time our regional patients are traveling for those studies. And so some studies that's practical and some it's not. Often on the studies being run by pharmaceutical companies, the company will reimburse you generously or appropriately for your travel and accommodation for participation in the study. So at least the cost aspect, but the time aspect, of course, you can't overcome. There is a lot of talk about teletrials and how do you bring these studies out to smaller centers. A lot of work is being done, and I can tell you my colleagues in regional centres in northern New South Wales are working extremely hard to make this a reality. And the ALLG, the Australasian Leukemia Lymphoma Group, is working very hard to make these a reality. But it is complicated, not just from a regulatory point of view. And it's simple things like if you're getting an infused experimental medicine, who makes that medication up in a regional centre in a small town? How do you get it there when it's stable? How do you administer it and monitor for side effects appropriately and things like that? So a lot of work is being done on this, but it's a very hard problem to solve. I agree. And so unfortunately, the best way is to participate in more studies, any studies that your local regional centre has, try and get onto them, prove that there are the patients in that region that need clinical studies. And that will help the doctors there in the clinical trial units there get more studies in the future. And it's about, building slowly, building capacity slowly, unfortunately, at the moment.
Thanks, Voigt. Thank you for your time today in presenting to us. I'm sure that the community got a lot out of it, as I mentioned earlier. I know that I sure did. So thank you very much for volunteering and coming to present in week 1 of National Myeloma Month.
Thanks for inviting me. Thanks very much.
We're going to head into a short break in a second, but I just wanted to remind everybody to please make sure that you complete our evaluation form, which is located at the bottom of our webpage. You can either do this during today's presentations, or the link will be available, still be available at the conclusion of the presentations today. We are now going to head into a short 5-minute break, and at the conclusion of this 5-minute break, one of our ambassadors, Sandy Roberts, we will be showing a short video from him. Enjoy the break. It's only 5 minutes, so make sure you get back for our ambassador, and then Kath will be introducing our 3rd presentation today.
Hello, I'm Sandy Roberts. You might know me best from television, commentating on sport from across the world, and right here at the home of sport, the mighty MCG. In this place, I've witnessed great triumphs, great wins when great teams and great athletes have had their backs against the wall. Myeloma Australia is a charity that provides critical support to people living with myeloma and their loved ones. 10 Australians every day get diagnosed with an incurable disease that most have never heard of. 27% of those will die within 18 months of diagnosis. Hopefully, very soon, this blood cancer, myeloma, will be curable. And medical professionals, our scientists and our nursing teams all believe a cure is within our grasp. This is my greatest call. This is me asking you for your help to cure myeloma.
Welcome back, everybody. It's my pleasure to introduce our third presentation. Ty Simpson is the CAR-T clinical nurse consultant for clinical trials at the Alfred Hospital in Melbourne. He coordinates care for clinical trial patients from the early stages of pre-screening, acute inpatient admission, and through to long-term follow-up. Ty manages patients with a variety of hematological malignancies receiving novel CAR-T therapies. He will also oversee the introduction of NK CAR cells for solid tumour in medical oncology. Ty began his career working in intensive care before following his passion into haematology. Ty's other interests include the patient experience, improving patient outcomes, and holistic care. Ty's presentation today is titled CAR-T, the Australian Patient Experience. Thanks for being with us, Ty.
Thanks, Kath, and thanks to everyone from Myeloma Australia for asking me to present today. It's really exciting to be able to talk directly to patients. Usually when we're giving chats, it's to colleagues and to referring specialists. So it's really great to be able to talk to patients and families and their carers today. So as Kath said, I'm the CAR-T nurse from the Alfred. I work in clinical trials, mostly looking into novel CAR-Ts, so CAR-Ts that haven't been given to humans before, but also looking at CAR-Ts that we know work in a certain population of people, but trying them in different populations. So my talk today hopefully runs for just over 20 minutes. I'm going to cover, it was previously mentioned before, that there's quite intensive screening for CAR-T cell therapy. So what workup and investigations you'll go through, how we collect the T cells, what chemotherapy we give prior to the CAR-T infusion, the CAR-T infusion itself, and the common side effects that you may experience when you're admitted or in the long-term follow-up period, the role of the carer and the role of the patient during follow-up, what we do at 28 days after the infusion to see if there's been any movement from the CAR-T cells, what's involved in long-term follow-up, and what life looks like after CAR-T cell therapy. So as mentioned before, there's screening and workup involved in CAR-T cell therapy. That usually is done over a couple of days, but I'll break that down in a little moment. The screening and workup investigations, once you meet all those criteria, we proceed with the CAR-T cell collection or the T cell collection prior to having them genetically reprogrammed to become killer T cells. The chemotherapy before the CAR-T infusion, the CAR-T infusion itself, and then follow-up. So screening investigations, they're usually completed over 1 to 2 days. Different hospitals will vary depending on the capacity, and the majority of the time, patients have this completed as an outpatient. So you won't need to be admitted for this. We look at the myeloma protein and inflammatory markers of the blood. We also check for different type of organ function. We perform a bone marrow biopsy looking for myeloma cells in the bone marrow. We look for myeloma protein in the 24-hour urine collection. We do cardiac scans to make sure the heart's okay for chemotherapy and some of the side effects that you may experience. and we perform imaging either via CT or a PET scan. We look at dental history, and we assess the veins to plan for the T cell collection. And at that time, if your veins have been exposed to chemotherapy before, you may not have good access. So it's just about planning for how we're going to collect those T cells, which I'll go through in a moment. One of the common not common side effects, but one of the side effects of CAR T cell therapy is neurotoxicity. So understanding what the brain looks like before we give CAR T cell therapy. And part of that's with a neurology review, either via a neuropsychologist or a neurologist. So apheresis is how we collect the T cells. It's also called leukapheresis, and is the process of collecting the T cells from this peripheral blood. Patients that have had a stem cell transplant before would be familiar with a stem cell collection. It's done either peripherally or via central line catheter, as I mentioned before. The thing with this is that it's called a non-mobilized collection. So when we're collecting the stem cells, we have to give a medication to get the stem cells out of the bone marrow and into the blood. But with the T cells, they're already circulating. When you're on the machine, there can be some changes to your blood pressure. And you have to remember we're removing part of the blood. So you can experience tingling of the lips or tingling in the hand. That's completely normal. We run transfusions during the collection. And you're observed by a specialist nurse, and it's one-on-one. The procedure itself takes 4 to 6 hours. It's again done as an outpatient. It's very boring, so please bring something to entertain yourself. So you can see here this patient's having apheresis product taken out. There's a draw line from one of her arms. It goes into this high-tech, what I call washing machine, over there with the lines on top of it. And so what happens is the blood comes into the machine, it spins very, very fast, and it separates all the different blood components. And from that, we can isolate and take the T cells. The blood is then mixed back together and reinfused to the patient on the other side. So it's usually either done in a chair or it can be done in a bed over that 4 to 6 hour period. So down at step number 1, it's leukapheresis, so collecting the T cells. The T cells, usually this process is over 1 day. Some products do require a 2-day collection if we haven't collected enough cells. And then the cells in Australia, because the manufacturing site for these cells are overseas, either in Europe or America, the cells are then frozen here and sent off for manufacture. And that's where the programming of the cell is hijacked, and they are reprogrammed with specific, in this case for myeloma, BCMA antigen, which is the attraction that they'll have on the myeloma cell. Once they've done this process, the CAR T cells are then expanded, and then they go through conditioning before they're sent back to us. And this process, generally, because we're so far away from where it happens, takes about 4 to 5 weeks. Because 4 to 5 weeks can be, or even sometimes 6 weeks, can be quite a while for someone to go without treatment, we sometimes look at something called bridging chemotherapy, which is basically us giving you some chemotherapy while the cells are being made to keep everything at bay. It can be, if you're referred, as previously mentioned, from a regional or a remote centre where you normally have chemotherapy. If you get sent to us for CAR T cell therapy, you can have this bridging chemotherapy done at home. It can include not only chemotherapy of tablets and intravenous or subcutaneous injection, it could be radiotherapy. And at the conclusion of the bridging chemotherapy, we do all those restaging assessments. And that's so that we have established a new baseline. And that 28 days after the infusion, we can check that the response that you've had is actually from the CAR T cells and not from the bridging chemotherapy. So the cells are back and we're ready to go. They've passed all their tests and we've got a product that we're ready to infuse. Depending on where you have CAR T cell therapy, at the Alfred, we give what we call lymphodepleting chemotherapy. So lymphocytes are the T cells that we're we collect to turn into CAR T cells. And what we do is we lower the lymphocyte count to allow for the CAR T infusion. Because what happens is when we take the T cells out, is your body replaces them quite quickly. And so we lower the T cell count, but by doing that, we can also lower some of the red cells and some of the platelets. So it's given over 3 days. It's given, we like to do it over a Wednesday, Thursday, and Friday. So we refer that as day -5 before the infusion to day -3, so 3 days. And it's given via central line, so through a PICC line or a Hickman's. And we use fludarabine and cyclophosphamide, which is generic. It's used for most lymphodepletion for CAR T products. Again, it's done as an outpatient. It's a 2-hour appointment. And because we're giving chemotherapy, we need to make sure that all your counts are robust. So we're looking at doing blood tests daily. And if there's anything low, we're replacing that while you're here. So by lowering your lymphocyte count, the T cells, they're part of the inflammatory immune response. You're at risk of infection. And one of the other white cells that we watch very, very closely, which everyone would be familiar with, is the neutrophils. And patients can become neutropenic during this phase. So there's 3 days of chemotherapy followed by the weekend. And during the weekend, the patients get to go back home. If you're referred to us, you would stay close by in local accommodation. Or if you live in Melbourne, for example, near the Alfred, you get to go home for the weekend and you represent on the Monday for the CAR T infusion. So the CAR T infusion is either done as an inpatient or an outpatient. We do it as an outpatient at the Alfred. We get you in nice and early. We do blood tests to make sure that everything's on track. We do what we call a baseline ICE assessment, which I'm going to go through shortly. We also do other complex neurological assessments. Now, the infusion itself is very, very quick. It only lasts about 15 to 30 minutes. Some of the products in clinical trials can be given over minutes. It's infused again through the central line, so no need for constant, blood tests and cannulas. You get the line in prior to the chemotherapy, and that stays with you right until day 28. We observe you very, very closely. It's very anticlimactic. Patients are sort of waiting for this big bang, but it's just as simple as sometimes for CAR Ts for myeloma, they're pushed through a syringe, and other times they go through a small bag through the drip. So the ICE neurological assessment, there's its breakdown. You don't have to remember that. Is an assessment that we do to test for neurotoxicity. So we ask you if you can tell us the year, month, and city. If you can tell us where you are, we get you to follow a command. It's a very simple command, as easy as touch your right ear with your left thumb. We point to 3 objects and get you to name them, something as simple as a cup, a pen, and a name tag. We get you to count back from 100 by 10s all the way to 0. And then we ask you to write a sentence. The sentence, we are looking for changes in the handwriting. It's very, very interesting. These questions and the assessment seem very, very basic, but these basic questions were able to pick up small changes in cognitive function and handwriting. Okay, so we all know Pac-Man. So you can see in the second G, there's some myeloma cells. There's a blue one, a pink one, a yellow one, and a red one, and Pac-Man's down the bottom. So these are charged CAR T cells. And I explain to my patients, it's like we've just put in 10 million Pac-Man and they know exactly what they're doing. They're going to find the myeloma, they're going to munch it down and hopefully get rid of it. At a closer look, you can see this is a very fancy picture, but how I'm going to explain it is the top yellow cell is a T cell, and this purple cell to the left-hand side is myeloma, or BCMA we'll call it. And what you can see here is that the CAR T cell is attracted to the myeloma and initiates the immune response, so the body's own inflammatory immune reaction to the myeloma cell. You can see there's the small white specks, which I'll refer to as cytokines. And the cytokines are then released into the blood, which is kind of the sort of ready, set, go of the immune system. So cytokine release syndrome, as I said, is the release of cytokines from the CAR Ts doing their job. It's experienced by about 95% of patients. It's very common. Most patients experience it in the more what we would call grade 1 CRS, which is just an isolated fever on its own. Patients may also experience low blood pressure, which we give fluids for, or temporary low oxygen concentration, which we give oxygen for. Now, so we watch you very closely, and at the first sign of fever, we initiate aggressive treatment straight away. We know that the harder we treat these things from the beginning, the less severe they get, and for a reduced duration. So some patients may only get a fever, some may get fever and low blood pressure, some may get fever, low blood pressure, and low oxygen, and may need a trip to ICU for blood pressure monitoring. We use, as I said before, it's an inflammatory response. We use anti-inflammatory medications, things like dexamethasone, but also we're able to slow down the immune system by giving special antidote medications. And the one for cytokine release syndrome that we use most commonly is called tocilizumab, which you don't need to remember. So neurotoxicity we refer to as ICANS. Everything's abbreviated in medicine, as you probably know. It stands for immune cell associated neurotoxicity syndrome. And so this is less common, but can be more severe. So we get you to do the handwriting test minimum once a day. And as things start to change, we increase the frequency. So when we're watching you write the sentence, we may notice a tremor in the handwriting, or the words may be misspelt. There may be some mild confusion. So you're alert and orientated, but may not be able to recall the exact date, or the day of the week or the year, or exactly where you are. There could be some fluctuation in mood. And in the more severe and very rare side effects, things like seizures and loss of consciousness. The way I think about it is that those cytokines from that picture before, the little salt granules that looked like they were being released from the T cell, is some of those inflammatory markers can cross into the brain, and it causes inflammation of the brain or swelling. And like CRS, we treat very aggressively from the moment we pick it up, again with anti-inflammatory medications. And anakinra is an injection that we give, which is a very fancy antidote that is quite effective at reversing neurotoxicity, or at least slowing it down. So what does this all mean? You'll be admitted on the ward by this stage. And when patients have a fever, how do we know if it's cytokine release syndrome or an infection? Because the cardinal sign of infection in a hematology patient is a fever, and the cardinal sign for CRS in a CAR T patient is fever. So we've got some investigations to do. So just because you've had CAR T and you get a fever doesn't necessarily mean it's CRS. We do what we call a septic screen. So we're looking for bacterial organisms all through the body, via the blood, urine. We might get you to cough up some sputum if you can. And we wait for these to come back. During that time, we treat you for both. So we've got a process of elimination. We treat you on what we call empirical cover for infection, but also treat you pretty aggressively for cytokine release syndrome at the same time. That's so that if in 3 days there's a bug there that we've picked up on one of the cultures, we're not 3 days behind. Because as I said, part of the lymphodepleting chemotherapy is about lowering those white cells, so you are at risk of infection. And once this is all happening, we monitor your temperature much closer. We increase our neurological assessments and increase the review by not only the CAR team, but our neurology colleagues as well. So we refer to the acute period as from the day of infusion all the way until day 28. The lymphodepleting chemotherapy is given as an outpatient from day -5 until day 0. It's only given over those 3 days, but we'll just count those days in over the weekend just to timeline things a little bit easier. As I said, it's as an outpatient, and the most common side effects is either the low blood counts, some nausea, lack of appetite, and low blood counts requiring some top-up. The inpatient monitoring stays from the day of infusion or some centres will admit the patient a couple of days after the infusion. It is in the inpatient setting. Inpatient setting is very, very busy. It's noisy. And I think talking to patients after they've had therapy, I think this is the most stressful time, because you're away from your family, you're away from your support people. You get us coming up to you and being like, oh, you've got a fever and we need to do all these tests. So it's very, very overwhelming. The most common side effect during this time is the CRS and the ICANS, low blood counts, and of course reviewed by all the hematology teams, but also the neurology teams as well. From about day 14 is when you've passed your highest risk period. And from there, we plan discharge education, not only with the patient, but the person that will be caring for them for those 2 weeks after discharge. And we call that a discharge education session. And during that 2-week period, you're required to stay within 1 to 2 hours of the hospital. We see you twice a week as an outpatient for relevant blood tests. We do those handwriting assessments, and then we obviously care for your central line as well. But one of the most important things is that we teach your carer how to do that neurological ICE assessment. So they're the person that's going to be responsible for giving us a call and saying, you know, my loved one's dropped a point on their assessment, or I have concerns for their cognition. What should we do? And that's what we go through in the education session. So the role of the carer is very, very important. We know that when we're considering patients for CAR T-cell therapy, one of the things particularly that we look for is that they have a carer that can be with them from day 14 until day 28. We teach you how to do the daily ICE assessment. We do temperature monitoring, so before each meal and before bedtime. We look out for the signs and symptoms of what to look for, so escalating to the medical team if you have any concerns or deterioration in handwriting. And then we have a transfer to hospital plan. So if your loved one has a fever at home or you're concerned about them, what that looks like, what's appropriate to be transferred in a car versus what's appropriate to be transferred by a ambulance. And of course, you're responsible for being in the same, I say house now, I used to say room, but you do not have to be in the same room together 24/7. So the role of the patient, we know that the highest risk period is in the first 2 weeks, but the risk for neurotoxicity can be up to 8 weeks. So there's no driving for 8 weeks. Please do not drink alcohol for the first 28 days after the infusion. It can be hard to differentiate between what's intoxication versus what's neurotoxicity in our assessments. So we just have that as an exclusion rule. Come to your outpatient appointments with your carer and bring all the documentation with you. And the important thing about bringing the documentation and your carer with you is that there's some small check-in and a bit of a handover as to how everything's going at home. So you've made it through to day 28 now, so it's 28 days after the infusion. We're going to retest for everything and check if there's been a response to the CAR T cells. So looking at the myeloma markers in the blood, urine and bone marrow. Repeat imaging. We look for ongoing CAR T persistence, which we can do via a blood test to see if there's any circulating CAR Ts. The big thing that I want to make clear here is that in other types of cancer that we use CAR Ts for, there's a big emphasis on the day 28 response, because the thought is for those cancers that if there's minimal or no response at day 28, that's the maximum response that's going to be experienced. In myeloma, we know that the day 28 assessment is not everything. And I've had patients that have had partial response or reduction in their paraprotein by 50%, which is pretty significant at day 28. And then at month 2, that comes down further. And they don't reach remission until month 6. But I just caught up with that patient 18 months ago and she's still remission free. So it doesn't mean everything, the day 28 assessment. From day 28, obviously we've played with your immune system, we've given you some low dose chemotherapy. You may require supportive care, so regular blood tests, neutrophil stimulating medication, and maybe a blood transfusion. We follow you up very, very closely. So on day 28, if you're stable, all patients are referred back to their primary hematology team. So if you come from the country, we refer you back if it's appropriate. If you come from interstate, we refer you back to your home team to follow you up really closely. And the primary care team then takes over. The follow-up that we have may be different to other hospitals. So I've just included what we do. We see you face-to-face every 28 days for the first 6 months. So it's every month you come and see us. And then after that 6 months, we see you every 3 months. So the frequency is reduced. So we see you every 3 months for 1 year. And then we follow you up via phone call or telehealth or the equivalent every 6 months for 15 years as a mandated requirement for follow-up after these sort of medications that play around with your immune system. So life after treatment, as previously covered, we don't really talk about the need for immunoglobulin replacement, but I think it's important to note when we target BCMA is not only present on the myeloma, it's actually present on some of the healthy antibodies as well. So these CAR T cells, they're attacking the myeloma, but they can also destroy some of the healthy immunoglobulins. So regular immunoglobulin replacement, either in the hospital or at home. During the first sort of 6 to 12 months, we put you on prophylaxis. So thinking things like valacyclovir and Bactrim, we revaccinate you against COVID and all your childhood immunizations. There's quite a robust plan around that. And then we would like to see some blood tests every couple of months, either from us here or from your referring center, depending on where you are referred to us from. And so if you're on the PBS, when CAR T is approved through the PBS, we will follow you up a little bit differently to clinical trials. But as said before, the clinical trials like to watch their patients a little bit closer. And so that's it from me. Thank you. And I was just, I'll leave this slide here just if there's any questions.
Excellent. Thank you, Ty. That was so informative. I'm sure our audience got a lot out of that, as I did as well as a nurse in this area too. We do have a couple of questions for you. I hope that's okay. The first one that came through says, can you collect stem cells and T cells at the same time?
It's a good question. The short answer is no. So when we plan for collecting, let's talk about T cells first. The T cells are already in the blood, so that's the collection, what we call non-mobilized collection. And to collect the stem cells, we need to mobilize them using GCSF. So filgrastim is the medication that we use. So if we tried to do both, we'd get a little bit of T cells, a little bit of neutrophils, and a little bit of stem cells. So it's a little bit hard. If you're on a clinical trial, say, for example, where some patients get transplant and some patients get CAR T, we would collect stem cells after cycle 2, as per most hospitals in Australia. And we would also collect the CAR T cells usually a little bit later. So around cycle 4.
Right. Okay. Thank you. And the other question, just relating to the side effects, in your experience, do you see less severe reactions now with the better surveillance, like we've learnt along the way?
Yeah, definitely. I think the CAR T landscape has changed dramatically in the last, I've been doing it for 2 years now in myeloma, and looking at what changes we use in treatment and how aggressively we treat our patients now compared to 2 years ago is incredible. So I suspect the reduction in significant side effects is probably because we're treating everything very seriously from the beginning.
And learning each time as we go.
Absolutely, absolutely.
Thank you, Ty. Thank you so much for being here today. We really appreciate your time and your slides. It was fantastic. Really appreciate
Thanks for having me.
Thank you. And that concludes our seminar for today. We hope you found this session informative and uplifting. A reminder, you are still welcome to send through any questions for any of our speakers today via our nurses email, which is nurses@myeloma.org.au. We would really appreciate your feedback, as we've mentioned. So before you log off, if you would take just a couple more minutes to complete the evaluation via the link below the video on our webpage. Our second seminar this month is coming up on Thursday, the 28th of March, and the theme is self-empowerment. We've got more great presentations to share with you then, including a panel discussing patient and carer experiences. Before we say a final goodbye, I'd just like to express a heartfelt thanks to all of our presenters today and throughout the month, and to the seminar organising team at Myeloma Australia, who have coordinated a fabulous program. A very big thanks also to Patrick and the team at CVP for their hard work and skill in production. Thanks so much for joining us. Have a wonderful day.